Greetings, students. In this lecture, we will start to talk about the body tissues, and first we'll start with the epithelial tissue. But first, what is a tissue? A tissue is a collection of cells. These cells have a common embryologic origin, usually, and these cells function together to perform a specialized activity. So this is the definition of a tissue. We have a collection of cells. These cells, usually, they are derived from the same embryological source. And these cells, they may differ from each other in appearance, they may differ from each other in some of their functions, but together, they act together to perform a specialized activity, which is the activity of that tissue. In addition to the cells, a tissue contains a substance that is present between the cells called the extracellular matrix. Now, before we continue, starting from this lecture, down here in the footnotes, I'm going to add sometimes some derivatives of words. What are the origins of some of the terms used in histology? These may be beneficial for you if you learn them. Now, body tissues can be generally divided into four main types according to type of cells and the amount and content of the extracellular matrix. So, according to these two criteria, the cells and the extracellular matrix, we divide body tissues into epithelial tissue, connective tissue, muscular tissue, and nervous tissue. This table shows the main features of each of these basic types of body tissues. So in the nervous tissue, the cells have intertwining, elongated processes. We have very small amount of extracellular matrix, and the main function here is transmission of nerve impulse. In the epithelial tissue, the cells are aggregated polyhedral cells with small amount of extracellular matrix, and the main function is lining and secretion. The muscular tissue, we have elongated contractile cells. The cells are long, with moderate amount of extracellular matrix, and the main function is movement. Finally, in the connective tissue, we have several types of fixed and wandering cells with abundant extracellular matrix, and the main function is support and protection. These are some of the main features of each of these body tissues. And as you can see, they are arranged according to the amount of the extracellular matrix. So the least is found in the nervous, and the most extracellular matrix is found in the connective. Of course, as we go along the course, we are going to talk about each of these types in more details. What are the features of the cells found in that tissue, and what are the contents of the extracellular matrix? So we begin with the epithelial tissue. What are the features of the epithelial tissues? What are the features that if we see them in a tissue, we can say that this is an epithelial tissue? First, it covers surfaces, lines cavities. So epithelial tissue either cover the surface of an organ or line the cavities of the organ. Now, as a result of this, it is in contact with another medium, air or fluid, for example. Because as you remember, as you know, when we say line the cavities, this means that this epithelium is in direct contact with the lumen of this organ. And if the epithelium covers the surface of the organ, this means that it is in contact with the cavity, with the body cavity in which this organ is found. This means that it is exposed to foreign bodies and chemicals. To endure these adverse effects, the epithelium has a rapid turnover. Now imagine with me the epithelium lining the stomach. Now this means that this epithelium is in direct contact with the lumen of the stomach. And as you know, the lumen of the stomach contains acid, it contains digestive enzymes. So these, and also we have food, of course, inside the stomach. These are all substances that could damage the epithelium. And therefore, this damaged epithelium must be renewed. The epithelium that lines the stomach, because it is exposed to these damaging effects, it must be replaced, it must be renewed very often. And this is what we call turnover. Turnover means time from the birth of a cell till the death of the cell. In the stomach, for example, it's about 7 days. So every 7 days, every week, we have a new epithelium in the stomach. The second feature is that epithelium is formed of sheets of closely packed cells. So the cells are closely packed with each other. They are aggregated. And not just that, but they are arranged in sheets. They are arranged in layers, one layer above the other. Now, since the cells are closely packed with each other, this means that the extracellular space between the cells is small. And this is why we have small amount of extracellular matrix in the epithelium, because the extracellular space is small. In addition, because they are closely packed with each other, the cells assume a polyhedral shape. They have a polyhedral shape, a geometric three-dimensional shape, columnar or cuboidal, for example. The cells are polar and are connected with each other and with underlying tissue by various types of complexes. We are going to talk about this point in more details in later slides, but what is the polarity of a cell? Polarity of a cell means that the cells have different regions, and these different regions of the cell have different features because they perform different functions. This is what we mean by polarity. The fourth point, the epithelium rests upon a sheet of extracellular matrix called the basal lamina. So the extracellular matrix is not just found between the cells of the epithelium, but under the epithelium, deep to the epithelium, the extracellular matrix forms a sheet upon which the epithelium rests, and this is called the basal lamina. So this is always the truth. Whenever we have epithelium, we have basal lamina under it. In addition, epithelia have a layer of connective tissue under them. So we have epithelium, under the epithelium we have the basal lamina, and under that we have connective tissue. For example, the gastrointestinal tract, we have the connective tissue under the epithelium called lamina propria. In the skin, the connective tissue under the epithelium is called the dermis. Finally, epithelial tissues are avascular, meaning that they lack blood vessels. There are no blood vessels in the epithelium. So how do they get their nourishment? Epithelium takes its nourishment by diffusion from underlying vascular tissues. The connective tissue that we have just said is usually found under the epithelium, is highly vascular, and the nutrients of the blood vessels of that connective tissue pass to supply the connective tissue itself, and by diffusion it goes to the overlying epithelium to supply nutrients to their cells. If we look at these two images, here we have a cross-section through the small intestine. This is the lumen of the small intestine. So this tissue here, which is lining the cavity, This is an epithelium. In addition, this tissue, which is covering the surface of the intestine, this tissue here, this is also an epithelium. This is also an epithelium. So the epithelium that lines the cavity is in direct contact with the lumen. And as you know, in the lumen, we have air, we have fluid, and this fluid contains enzymes. It may contain toxic substances, it may contain microorganisms, and this is why we said that the epithelium may be exposed to damaging factors and must be replaced. Under the epithelium here, we have a connective tissue. This is the lamina propria. Also, this part here, under this epithelium, we have also a connective tissue. What about this image here? This is the skin. This is the epithelium of the skin. And this is called the epidermis. The epithelium of the skin is called epidermis. And this is the connective tissue found under the epithelium. And in the skin, it is called the dermis. Of course, remember that the epithelium of the skin, the epidermis, is exposed to the external environment. And as you know, in the external environment, we have heat, we have cold, we have objects causing trauma to the skin. So this is why the epithelium must be replaced often. What are the functions of epithelial tissues? There are many functions. Here we are going to talk about general functions because the function of the epithelial tissue depends on its location. The general functions of epithelial tissue include lining, covering and protection, secretion, for example, the epithelium of the stomach, and the glands, because glands are considered epithelial tissue. Also absorption, for example, the epithelium of the intestine. And finally, contraction. We have a type of cell, a type of epithelial cell called myoepithelial cell. These perform contraction, and we are going to talk about this later on. Now we're going to talk about two important definitions, basal lamina and basement membrane. Basal lamina is a sheet of extracellular matrix located under the epithelium. As we have said, all epithelium have basal lamina under them. It is a very thin layer and can only be seen by the electron microscope. So here we have, in this image, this is the epithelium. These are the epithelial cells. As you can see, they have a geometric shape. Also, you can see that the intercellular space between them is very small, cannot be seen in this image even. Under it, here, this is the basal lamina. Basal lamina. Basement membrane is a much thicker structure seen by the light microscope. It is formed of the basal lamina and the reticular lamina. We know what basal lamina is, but what is the reticular lamina? The reticular lamina is the upper reticular fiber-rich part of the connective tissue that is usually located under the epithelium. As we have said, we have epithelium, we have basal lamina, and under the basal lamina, we usually have connective tissue. So this is connective tissue. The upper part of the connective tissue found under the epithelium, this region here, this part here, this is rich in a type of fiber called reticular fiber. So in this part, we have a lot of reticular fibers, and this region is called the reticular lamina. Reticular lamina. The basal lamina with the reticular lamina together form what we call the basement membrane. Basement membrane. So these two images, in this first one here, this is a transmission electron microscope image. This is a cell, this is a cell, And here we have the cell membrane. This is the cell membrane. Notice how a thin white line surrounded by two thin black lines. Under it we have the basal lamina. The basal lamina. Here, this is the basal lamina, formed of extracellular matrix. And under them, as you can see here, this part, we have an area containing many reticular fibers. All these are reticular fibers. So this is the reticular lamina. This is the reticular lamina. Together, they form the basement membrane. In light microscope image here, the red lines indicated by the arrows, these are basement membrane. All these are basement membrane. So in the light microscope, when we see a thin line under the epithelium, in light microscope, when we see a thin line under the epithelium, this is the basement membrane. This is the basement membrane, which is composed of the basal lamina and the reticular lamina. What is the importance of the basal lamina? What are the functions of the basal lamina? They provide structural support for the epithelium. They help in filtering of substances that pass through. Sometimes we may have holes in the basal lamina and sometimes we do not. So depending on the number and size of holes in the basal lamina, the substances that can pass are controlled. Also, basal lamina affect cell proliferation, differentiation, and migration, and they are important for cell repair, as in repair of nerve fiber and the neuromuscular junctions. When these two repair processes occur, the basal lamina must remain intact because they assist in the repair process. Types of epithelium. Epithelium can be divided into lining or covering epithelium, which line cavities or cover organs, and glandular epithelium. Glandular epithelium, the main function is secretion. However, we must remember that some lining epithelial cells secrete, for example, some of the lining epithelium of the stomach. And some glandular cells are actually found between cells of the lining epithelium. For example, goblet cells. Goblet cells are glandular epithelium, but they are found lining the small intestine, for example. We start with the lining or covering epithelium. How do we classify this epithelium? We classify this epithelium by two steps. First, according to number of layers. How many layers are there in the epithelium? Remember we said that one of the features of the epithelium is that the cells are arranged in layers. So how many layers are there? If the epithelium contains one layer, it is called simple. If the epithelium contains multiple layers, it is called stratified. Now pay attention, when we are talking about epithelium and we say multiple layers, we mean that we have two or more layers. So if an epithelium is formed of 2 layers, it is considered stratified. If it is formed of 20 layers, it is considered stratified. So according to number of layers, it is either simple or stratified. Then we come to the simple epithelium, which is formed of only one layer of cells. We ask ourselves, what is the shape of this epithelium? What is the shape of the cells in this epithelium? This could be either squamous, cuboidal, or columnar. So this means that the simple epithelium could be either squamous, cuboidal, or columnar. When we come to the stratified epithelium is formed of multiple layers. So we ask ourselves, in the second step, we ask ourselves, what is the shape of the cells in the topmost layer of this epithelium. Now, what do we mean by the topmost layer? So if I have so here's an epithelium. I have two layers. This is one layer and this is the second layer. So this is a stratified epithelium. And this is the basement membrane or the basal lamina. This is the basal lamina. As we said, all epithelia have basal lamina under them. So I have two layers. One of them is directly resting on the basal lamina, and the other one is above this layer. So the topmost layer is the layer farthest away from the basal lamina. The layer farthest away from the basal lamina, this is considered the topmost layer. This is considered the topmost layer. And when we classify stratified epithelium, we look at the shape of the cells in this layer, the topmost layer. This could be squamous, cuboidal, columnar, or transitional. And the squamous, the stratified squamous, could be keratinized or non-keratinized. So these are the types of the stratified epithelium. And we have another epithelium called pseudo-stratified epithelium. This has features of both stratified and simple, and we are going to talk about this one in the next slides. So we start with the first epithelium, the simple squamous epithelium. It is formed of a single layer of flattened squamous cells. This is how we define epithelium. First is the simple, so it is formed of a single layer. The type of cells is squamous, which means that it is squamous cells. Squamous cells are flattened as we are going to see them. Where do we find this type of epithelium? Now, we must pay attention. Epithelium is found throughout the body. We are, of course, not going to take all types of epithelium all over the body. We are going to take some examples here. You are going to study the different types of epithelium in different organs as you progress through the years in your med school study. So here, in this course, we are only going to talk about some examples. So the simple squamous epithelium could be found in capillaries. Here in the capillaries, the simple squamous epithelium is given a special name, endothelium. Endothelium. Also, simple squamous epithelium is found lining of body cavities. The cavities that are found in the body are the pericardial, the pleural, and the abdominal cavities. These are all lined by simple squamous epithelium, and here it is called mesothelium. Mesothelium. Also, the alveoli of the lung are lined by simple squamous epithelium, and these are called pneumocytes. Pneumocytes. What are the functions of simple squamous epithelium? Again, the function of the epithelium depends on the site. Here we are going to talk about the function generally. So the general function of the simple squamous epithelium, because they have a thin cytoplasm, they allow various substances to pass easily across them. Now, squamous cells are thin, flattened cells. So if we try to draw a squamous cell, so here we have so this is the squamous cell. This is the cytoplasm, and bulging from one side is the nucleus. As you can see, the cytoplasm is very thin, very thin. Because it is thin, substances can easily pass through the cytoplasm. This is why we find this type of epithelium in capillaries and in the alveoli. Remember, in capillaries, we have exchange of nutrients and waste products and gases. And also in the alveoli, we have exchange of gases. So because we have exchange of materials and a lot of material are passing through the cells, this is why we have simple squamous cells, because of their thin cytoplasm. Now, for mesothelium, in addition, mesothelial cells perform another function, which is secretion. They produce a lubricating fluid that reduces the friction in body cavities. So if we look at these images, this is the epithelium, simple squamous epithelium. As you can see, this is the basement membrane. This is the basement membrane, and this is the underlying connective tissue. Notice how the cytoplasm is thin. The cytoplasm of these cells is thin. And if you look at this image here, this is a section through the alveoli of the lung. So here we have an alveolus. If you notice, the wall is very thin. The wall is very thin because it is formed of simple squamous cells, simple squamous epithelium. The red line of the wall represents the cytoplasm of these cells, whereas the dark dots, the small dark dots that we see, these are the nuclei of the squamous cells. Simple cuboidal epithelium. This is an epithelium formed of a single layer of cubical cells. As you can see here in this image, the cells are cube-like. Now, we find them in the renal tubules and covering the ovaries. This is a renal tubule. This is the lumen of the renal tubule. And here we have the cuboidal cells of the epithelium. And here we have the basement membrane, this thin line, this thin line here. This is the basement membrane. Now, as you can see, we have only one layer of cells, so it's simple cuboidal. It is usually found covering some organs like the ovary, and it is involved in active transport. So this is important. This is one of the first relations between appearance and structure. Cells that perform active transport are usually cuboidal in shape, and cells that are cuboidal in shape usually perform active transport. Simple columnar epithelium is formed of a single layer of tall cells that could be ciliated or not. So here we have the basement membrane, and notice how the cells are tall. They look like columns, and this is why it is called columnar cells. Now, this type of epithelium could be covered by these hair-like structures, cilia, or it could be not covered. In this half of the drawing, here we have non-ciliated simple columnar epithelium. So the ciliated simple columnar epithelium is found in the uterine tubes. The non-ciliated simple columnar epithelium is found in most of the gastrointestinal tract. For example, in the stomach, in the small intestine, in the large intestine, and, for example, in this image here, this is the gallbladder. This is the gallbladder. So these are the columnar cells, and we have only one layer. So this is simple columnar epithelium, and on the surface we do not see cilia. This is the surface of the epithelium. We do not see any hair-like projections, so it is non-ciliated. Now, the function of this type of epithelium, it could be secretion, for example, in the stomach, or absorption in the small intestine. The stratified epithelium. We must remember that stratified epithelium is an epithelium formed of 2 or more layers. So we start with the stratified squamous epithelium. Stratified squamous epithelium. And we start with the keratinized type. So stratified squamous keratinized epithelium. So stratified means that it is formed of multiple layers of cell. Squamous means that the topmost layer is formed of squamous cells. Keratinized means that the epithelium is covered by keratin, which is a layer of non-living material. Now, this is found in areas that require great protection, for example, the skin, in which it is called the epidermis. The stratified, squamous, keratinized epithelium of the skin is found in the epidermis. The functions of this type of epithelium include protection, and a very important function for this epithelium is prevent water loss. Prevent water loss. This is very important. So if we look at this epithelium, this is a section through the skin. This is the epithelium. This is the layer of keratin. This is the layer of keratin, which is formed of non-living material, but it is actually part of the epithelium. And if you look at this diagram, all this is the epithelium. As you can see, it is formed of multiple layers. This is a layer, and here we have another layer, and many, many layers. All these are layers of this epithelium. And as you can see, this part here have no nuclei because the cells here are dead, and this is what we call the keratin layer. So this is stratified squamous keratinized epithelium of the skin, which is called the epidermis. Stratified squamous non-keratinized epithelium. Again, it is an epithelium formed of multiple layers of cells. The topmost layer is formed of squamous cells, and the epithelium is not covered by keratin. It is found in areas that require protection, and water loss is not a big problem. Remember one thing: the more the organ requires protection, the more layers of epithelium there will be. Okay? So where do you find this type of epithelium? It's found in the mouth, in the esophagus, and the anal canal. It's also found in the vagina. In all these areas, we require great protection. However, water loss is not a big problem because in the mouth, the esophagus, and anal canal, any water in the mouth and the esophagus, for example, any water secreted is eventually reabsorbed. So it is not a very big problem. Here in this image, this is the epithelium. It is formed of multiple layers. All these are layers of cells. Notice how in this layer we have some kind of columnar cells. In this layer here we have polygonal cells, different shapes of polygonal cells. As we go up, the shape of the cell changes, and here we have squamous cells. Here we have squamous cells. So the different layers of the epithelium have cells with different shapes. So when we classify epithelium, we only look at this layer, the topmost layer. Why? We decide this is the topmost layer. This is the basement membrane, or the basal lamina of the epithelium. So the layer, this layer, is the furthest from the basal lamina, so it depends on the shape of the cells in this layer. Again, this is the epithelium, and these are the topmost layer, squamous cells. And since we can see nuclei in all the layers, this means that this epithelium is not covered by keratin. So this is stratified squamous non-keratinized epithelium. And now we'll talk about stratified cuboidal and columnar epithelium. These are less common than the other types, and the stratified cuboidal, of course, have multiple layers. The topmost layer is formed cuboidal cells, as you can see here. Here we have one layer, and here we have another layer, and this is the basement membrane. Usually we have two layers, it could be more, but what's important is that the topmost layer is cuboidal. It is cuboidal. It is found in large excretory ducts of salivary and sweaty glands, and the function is protection and secretion. Stratified columnar, also multiple layers. The topmost layer is formed of columnar cells. So here is the basement membrane. This is one layer, and this is another layer. What we care about is this layer here, the farthest away from the basement membrane, and it is formed of columnar cells. It is found in the conjunctiva, and its function is protection and secretion. What is the conjunctiva? The conjunctiva is a thin membrane that covers the sclera of the eye. The white of the eye is covered by conjunctiva, and it also lines the inner surface of the eyelids. It is a protective membrane. So here in this image, we have the large excretory ducts. So this is a duct. This is the lumen. This is a cross-section through the duct. This is the lumen. Here we have the first layer of cells. And here we have another layer of cells, which means we have two layers of cells. And the topmost layer, this is the basement membrane. This is the topmost layer. The cells here are cuboidal. So this is stratified cuboidal epithelium. This image is stratified columnar epithelium of the conjunctiva. Transitional epithelium, also called urothelium. This is a stratified epithelium. Stratified epithelium. Notice how we have multiple layers of cells here. The topmost layer of the stratified epithelium are dome-like. So here's the cell, and the upper surface of the cell is dome-like. It is convex. And these cells are called umbrella cells. Umbrella cells. It is found in the urinary bladder, the ureters, and the renal calyces, which are all organs related to the urinary system, and therefore this is called urothelium, the epithelium of the urinary system. Now, why is it called transitional? The umbrella cells are dome-shaped when the bladder is empty. If we take the urinary bladder as an example, and we examine it, we'll see that the umbrella cells, the topmost cells of this epithelium, these cells, are dome-shaped when the bladder is empty. Once it is full, these cells will become flattened. They will stretch out with the wall and become flattened. So the shape of the topmost cells will change. This is why it is called transitional epithelium. The function of this epithelium is protection against the adverse effects of urine and allow the bladder to change size. Look at these two images. This first image here, we have the bladder when it is empty. Now when it is, notice that when the bladder is empty, notice the umbrella cells are dome-shaped. These are dome-shaped cells. This is the epithelium. This is the epithelium. So the cells are dome-shaped. Now what happens when the bladder is filling with urine? The wall will stretch. So the wall will be stretched in all directions. The epithelium will stretch with it. The epithelium will stretch with the wall. And these cells, the umbrella cells, will also stretch. They will also stretch. And instead of being dome-shaped, when we stretch them like this, they will become flattened. They will become flattened, as can be seen here. So this image represents the wall of the bladder when it is full. So this is the epithelium. Notice the topmost layer is formed of flattened cells. This is why it is called transitional, because the shape of the cells of the topmost layer changes according to the state of the organ. Now we talked about the simple epithelium, we talked about the stratified epithelium, and we talked about their types. Now we have a special type of epithelium called pseudo-stratified epithelium. So it is a stratified epithelium, but it is pseudo. Pseudo means false. So it is a stratified epithelium, but this stratification is false. What do we mean by that? In this epithelium, the cells have different heights. All cells rest on the same basal lamina, but not all of them reach the surface. This makes the nuclei occupy different levels, giving the epithelium a false stratified appearance. So this is the epithelium. Notice this is the basement membrane or the basal lamina. I have this type of cell here, a short cell. I have this type of cell, intermediately long. And I have this type of cell which is very high, reaching the surface. Notice that the three types of cells, all of them are resting on the basement membrane or the basal lamina, but only the tall one is reaching the surface. The other two, the short and the intermediate one, do not reach the surface. But if I look at the nuclei, I can see that they occupy different levels. For example, I have this level of nuclei, I have this level of nuclei, and this level of nuclei, and this level of nuclei. So actually, the nuclei occupy different levels. When I examine a tissue under the microscope and try to identify the epithelium as simple or stratified, I actually depend on the nuclei rather than the actual cell, because cell membranes do not appear clearly or sometimes may not appear at all in the microscope. So I depend on the nuclei. So if I see the nuclei occupying different levels, I say that this is a stratified epithelium. But since here all the cells are resting on the same basal lamina, this means that this is not a true stratification. Because when I say stratified, I mean I have a layer of cells. For example, here I have a layer of cells. Above that, I have a new layer of cells. And above that, I have a new layer of cells, and so on. This is what I mean by stratified epithelium. I have one layer above the other, one layer above the other. But in this case, the cells all are lying on the same basal lamina, which means all the cells are on the same level. But because they have different heights, their nuclei occupy different position, giving them a false stratified appearance. So if you want to be scientific about this epithelium is actually simple because it is formed of a single layer of cells. So it is simple epithelium but has a stratified appearance under the microscope. This is why it is called pseudo-stratified epithelium. There are many areas in the bodies with pseudo-stratified epithelium. One of them is the respiratory epithelium. Respiratory epithelium is a pseudostratified columnar ciliated epithelium found in the trachea, bronchi, and nasal cavity. The main function is protection and secretion. The ciliary movement of this epithelium remove particles from the airway passages. If we look at these two images of the respiratory epithelium, we see we have several types of cells, but not all of them reaching the surface. We have these cells. These are the columnar cells. These are the most common cells in this type of epithelium. This is why it's called pseudostratified columnar epithelium, and because it is covered by cilia, it is called ciliated epithelium. Here is another type of cell, but is intermediate cell, does not reach the surface. And here we have a small cell, does not reach the surface also. And here we have a glandular cell which secretes mucus. This is a gland, goblet cell. So this is a diagram showing the respiratory epithelium, pseudostratified columnar ciliated epithelium. If we look at the image here, this is how it appears under the microscope. Notice how I can see the nuclei occupying different levels. So by first glance on this slide, I will say, okay, this is stratified epithelium. But when they examined this epithelium by more methods, it turns out that actually all the cells are located on the same level. Also, I can see goblet cells. These are goblet cells. These are glandular cells that secrete mucus. And these are the cilia found on the surface of the epithelium. So this is the respiratory epithelium.